So get advantage of this flash talk. If you have any questions, you can ask after the talk or during the talk also. So welcome, madam, and floor is yours.
Dear students, actually we have scheduled the lecture as per the manual earlier. Besides that, we added some more flashbacks so that to have some kind of interaction with the specialist. Dr. Amrita Das is doing an other work on the yeast, especially on the antagonistic yeast. So the genomics of the yeast, antagonistic yeast and other thing will give a new dimension to the other whatever you have covered for the either the insect genomics or pathogenomics. In this lecture, we are benefited by her lecture. The floor is yours, Amrita Das.
Thank you, sir. So, am I audible? Okay. So, today we are going to have a talk on beneficial antagonistic yeast. So, mostly we are familiar with the yeast that are beneficial for the brewery, baking, wine making parts. So, little is we are less familiar with the yeast as an antagonistic microorganism. So, before going to the talk, we should know that what is yeast. So, you know what is yeast? You have a basic idea, right? So, it is a eukaryotic microorganism that is single celled and basically 3 to 4 micrometer in cell size, and it reproduces by mostly budding, and some of the yeast they can reproduce through fission also. And most of the yeast they belong to the Ascomycetes group, and some belongs to the Basidiomycetes. So we basically through yeast we mean the budding yeast, that is the breakery yeast we know, and they known to grow with a temperature range 25 to 30 degree centigrade temperature, and they basically love the acidic nature to grow. So whenever we go for the antagonistic isolation, so we prefer the fruits, right? Fruits are acidic in nature. So as I told, most of us familiar with the biocontrol agents, that is trichoderma, that belongs that is a fungus, and in case of bacteria, that is bacillus, pseudomonas. So these are the most common, right? We know. So we are less familiar with the antagonistic yeast. So why we should consider yeast as an antagonist? So there should be certain characteristics that we can take it as a antagonistic in nature. Like as I told you, yeast are single celled and they are prolific in growth, means they can grow rapidly, and they can grow in adverse conditions also. They require less nutrients, okay? So that in adverse nutrient conditions also you can able to grow the yeasts. So many yeasts has been exploited, and they has been mostly antagonistic yeasts are, we select the fruit surface for the isolation of the antagonistic yeast. As I told you, they are acidic, love the acidic nature for the growth, right? So for the isolation we prefer the fruit surface, but not only fruit surface, they can grow everywhere. So we can select the leaf as well as the soil, water also for the isolation purpose. So several kind means method we follow for the isolation, that if you go for the fruit or leaf, you can go for the leaf impregnated method, that we can directly put the leaf on the media, as well as we can take the washing water, means suppose if you take the fruit or leaf, you can wash it and you can use that washing water for the isolation of yeast purpose. And if you compare with the other fungus, other biocontrol agents, it grow rapidly, means like a bacteria, like for 24 hours you can have the growth of the yeast, okay? So the most well known antagonistic yeast is the Saccharomyces, right? So besides Saccharomyces, there are other antagonistic yeast that are being exploited, and they are using to study against the basically the post-harvest pathogens. Like you can find that Candida, Pichia, Rhodotorula, Hanseospora, then Aureobasidium. These are others, means you can say the non-conventional yeast, because Saccharomyces yeast is the conventional yeast, it is called the baking yeast. Besides Saccharomyces, we call this the non-conventional yeast that are also used as the antagonistic yeast, and they can be exploited against the many post-harvest pathogens, like you can exploit against the Penicillium, Botrytis, Colletotrichum, Aspergillus alternaria, mostly appear as a post-harvest on the fruits and vegetables also. So why we can employ the yeast as an antagonist? What make them to act as the antagonist? What are the basic antagonistic mechanisms we have heard? Can you tell me? For other biocontrol agents, is it really different from other biocontrol agent, or it is the same only? Same only. Mostly in case of other biocontrol agent, we will find the microparasitism, competition. Okay, so for yeast also, mostly the antagonistic mechanisms are same only, but yeast, the antagonistic way, the way they means act, it's a combined way. They not only single activity they can take up. Even if they go for the competition, they can also produce a secondary metabolite. Okay, it is a combination of mechanisms they employ to act as a antagonistic microorganisms. Like, okay, so what are the basic microbial means antagonistic mechanisms they employ? That is the competition. Basically, as I told you, they are rapidly grow very rapidly. So when they go for the competition, they can have the excess amount. They can intake the glucose as well as nitrogen, which make them means the causing an organism are not able to survive because the non-availability of the nutrient. Okay. So besides competition, they also act the microparasitism, but it is less known for the yeast, and mostly the action they employ is the production of the secondary metabolites. That is the volatile metabolites, that may be the killer toxin, that may be the secreted enzymes. Okay. So these are the major action mechanisms that make them to act as the antagonist. So there are several registered biocontrolled yeast products are there. So you can have a look. But since 2009 to 2021, generally 163 patents has been registered, but mostly around 53% are from other countries like Germany, USA, Australia and China. From India or from Asian country, it is very less. So since now the yeast are being exploited to act as a microbial antagonist. So for that purpose, exploration is very much essential. So we learned that the antagonistic activity of any biocontrol agent that is live or that is governed by certain genes, right? So to study what are the genes actually employed between against this to govern these biocontrol mechanisms, we should know the genome. So genome is not about only the whole genome sequence. Okay, it is the whole omics. What is omics? It is the whole learn about the genome sequences, then transcriptome, proteomes, as well as metabolics, right? So whole sequence or omics data only you have, then only you can tell, okay, these genes are responsible for this biocontrol activity. So in case of yeast, as I told, Saccharomyces cerevisiae was first identified as the biocontrol yeast in 1960. Okay, when scientists found that Saccharomyces cerevisiae could produce some killer toxin that can hamper or that can be antagonistic to other yeast. Okay, but that is not killing the itself Saccharomyces cerevisiae. So since then, to take up that yeast as a biocontrol agent has been started. Okay. Okay. Then the Saccharomyces cerevisiae was the first genome sequence that was studied, and the strain was S288C. The genome sequence was started in 1996. Till now, it is the only full, completed and well characterized genome sequence that are being used as a reference genome. Do you know what is reference genome? So reference genome is that right now or till now, whatever the genomes are sequenced for the eukaryotic organisms, they are being compared with the Saccharomyces cerevisiae with this strain only. So that we can study, we can learn what are the evolution, what are the genetic evolution has been going through. So after that, According to the means with line of this, right now there are 1,002 genome project is going on. So in this lab they have started to collect 1,000 yeast genome from different habitat, different regions, so that it can be annotated and compared against the Saccharomyces cerevisiae reference genome, so to study the evolutionary progress of the yeast. So here at the database, where you can go through the this database and you can take up the yeast sequence, and you can also annotate, or you can compare your genome sequence of yeast with this database. You can take up from here also. So after that Saccharomyces cerevisiae, several genome sequences has been done, but recently Candida oleophila strain 182 has been sequenced, and it is the first yeast-based product. It was used as a first yeast-based product, that is called the ICAR, and when it was compared with the available strain or the reference genome of the Saccharomyces cerevisiae, it was found that almost genome size was almost same, and when the gene annotation of the comparison of the gene annotation was done, it was found some important or evolutionary genes were found in case of functional genomes, and which could be employed for the in case of metabolic pathway, like specific protein genes that poses the NADH dehydrogenase activity and glutathione peroxidase activity was observed in case of Candida oleophila when compared to the I22. And it could means tell that it has definitely evolutionary processes going on, so that it has make it as a one of the potent antagonistic yeast against, and it can be employed against many post-harvest pathogen like Aspergillus penicillium also. So as I told, the genome study of genome is not only to study the gene only. Okay, you should go through the other parts of the genomics, that is the proteomic approach, metabolomic approach, as well as transcriptomic approach. So all these, like through genome sequence only you can annotate only you can know the genes. This much of genes are present in this whole genome. Okay, then only you have to annotate the gene to know their functional characteristics, like which gene is responsible for their particular microbial activity or antagonistic activity. Then only you get to compare or establish your antagonistic activity of that particular microbial antagonist. Okay, so in a whole holistic study, it will help to get to know, you can establish the antagonistic property of any kind of yeast, not only yeast, for any kind of microbial antagonists. Okay, so lastly I could say that, so this is a short flash talk only, so I could discuss with you whatever you are having in your mind. The exploitation of microbiome is very much necessary. Okay, so whatever we have, we can't say that it can be act as a microbial antagonist. So to get a potent microbial antagonist and to get a commercialized microbial antagonist, these two are different. Okay, once you have a potent microbial antagonist, but it may not be, you are not able to get it commercialized, right? There is the vast means road is there, you have to cross the road, because you have to make the antagonist more stable, you have to make the antagonist cheap also, you have to make the antagonist viable also. Until unless these things are there in your antagonists, you are not able to commercialize it. Otherwise you can do lots of work in your lab, you can search many microbial antagonists, basic work is going on, but these things are very much necessary to make it from lab to commercialization. Okay, so for this purpose, exploration of microbiome is very much necessary. Until unless you explore it, you will not able to get a potent one, and then you will not able to get the commercialized one. Okay, so if you have any queries, we can discuss. So anybody working on biocontrol agent? All are MSc or PhD? Both. So you must be working on a biocontrol agent only. No. No. Nobody is working on biocontrol. You are working on which? That. Yeast only. So have you isolated any from which habitat? Soil only. Phyllosphere. Okay. So against which pathogen you have tested it? Mostly post-harvest pathogen only. So how is the antagonistic activity? So do you find any relevance with this talk? Huh? Which kind of activity you have noticed? Means lots of mechanisms is there. Na? I told that not a single mechanism is employed for the biocontrol activity. Okay. So which kind of activity you have noticed? Secondary metabolites. Mostly they are a prolific producer of the secondary metabolite. So that is that makes them, that quality makes them to be more efficient as a antagonistic. They produce volatile metabolites. Non-volatile metabolites are less common, but mostly volatile metabolites are very much active in case of yeast. You can get lots of aldehyde ketone group. You can search any literature, and these groups are having a biocontrol activity or antagonistic activity, or they can control the many plant pathogens. So any question? No question means no understanding, huh?
How many heard about antagonistic yeast before this class? No one?
For what purpose you heard the name of the yeast?
Only two, huh?
Baking.
So this is another domain. It's not about the microbes, not about the insect, not about the pathogen. It's another domain where it can be utilized for the many purposes.
Ma'am, can I use this secondary metabolites to use for green synthesis of nanoparticle?
Nanoparticle, definitely you can prepare the nanoparticle. I have to extract the secondary metabolite. But again, I am talking about the secondary metabolites, and not a single metabolite you will not get. And suppose if you are going to extract the secondary metabolite, a lot of chemicals or substances will be there. Okay, so then again you have to select which metabolite, means among those chemical substances, which one is active. So there are certain in agriculture chemicals, actually they do column chromatography.
This metabolites used for profiling or HPLC.
Yes, you can do it. So you can get to know that what are the chemical substances are there only, but for to get to know which substances is actually active, you have to test each and every one.
So nanoparticle in general culture filter will work for the other function. So since it is also fungi,
You can able to get.
So like there is not magic of anything. All the fungus can be able to produce the nanoparticle. Which whether it's pathogenic, whether it's antagonistic, nothing is there. Because it's like enzymes what is produced by the fungi. It's all fungi is producing. So we have already worked a lot for the pathogenic. Any difference will be antagonistic fungi or pathogenic fungi. There's no difference. Definitely. Only thing is that maybe more precise, more that size will be more like less in some case, other case, but definitely can be. Okay, no other question. So, Bhaiya, can. So thank you, Amrita, for this. You go there.
Yes.
Okay, you have to take this background only. Background is necessary.
Thank you.
So madam is sitting in pathology with ma'am only. I'm working in ma'am's group only.